Hello, everyone. My name is Olivia Long, and I will be your moderator today. I'm absolutely thrilled to welcome you to today's webinar titled Advancing Oral Drug Development with In Vitro Dissolution and Absorption Tools. We have an incredible session planned for you all today, filled with insightful discussions and valuable takeaways. Before I get started, I'd like to extend a very warm welcome to our esteemed guest speakers, Carl Box, Chief Scientific Officer at Pion, and Balint Cinco, VP of Development at Pion, who will be joining us for the live Q&A. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the left of the auditorium and click to open the Q&A window. So, without further delay, I am delighted to hand over to Carl Box and would like to thank you for presenting to us today.
Welcome everyone. In this talk, I will present the latest work using Pion's in vitro dissolution and absorption tools for advancing oral drug development. Pion Inc. is a company that manufactures instrumentation and provides services for in vitro drug characterization. We provide expertise and tools for understanding phys-chem, dissolution, and drug absorption processes, and design instruments for studying both drug substance and drug product. Our headquarters are located in the United States in Billerica, Massachusetts, where we manufacture and assemble all of our instrumentation. We also have additional offices in the UK and in China. The UK site is our Centre of Excellence for contract research services. This slide shows the main steps involved in oral drug absorption. Drugs must be in solution before they can be absorbed across the membranes of the gastrointestinal tract. Hence, they need to have good dissolution and solubilization. Drugs also need to have good permeability characteristics to cross the membrane barrier and to be able to reach the bloodstream to be able to elicit an effect and show efficacy. In the next slide, I'll introduce the first of our absorption tools. This is the parallel artificial membrane permeation assay, or PAMPA, which is used to measure the permeability of active pharmaceutical ingredients. Pion was the first company to commercialize the PAMPA technique. We worked directly with the inventors at Roche in Switzerland. The assay is a high throughput 96-well plate technique for rank ordering permeability of APIs arising from drug discovery programs. Pion further optimized the original assay to demonstrate a good correlation with human jejunal permeability results. The Pion method shown on the left consists of a donor plate at intestinal pH separated from an acceptor plate at blood pH by an artificial hydrophobic membrane simulating the gastrointestinal tract walls. Compounds are dosed into buffer from DMSO stocks on the donor side, and the concentration reaching the receiver plate is measured after a defined time interval to determine permeability values. The calculations also take into account the pH gradient and membrane retention. Results can be readily displayed in the software using a plate map view. The middle figure shows 9 wells out of 96, where high permeability is shown in green and poor permeability of the drug is shown in blue. The right-hand graph shows the correlation with human jejunal permeability results. Once candidate molecules have been selected from the drug discovery process, the next steps are to assess drug formulation and drug delivery strategies. In this section, we will discuss some of these concepts. Many of the drug molecules coming through pharmaceutical company pipelines are poorly soluble compounds. This presents a problem with regards to overall drug absorption and may necessitate the use of an enabling formulation technique to enhance solubility. Drug molecules can be classified as different compound types. Greaseball molecules are oily with low melting points, high log P values, and an aversion for water, making them hydrophobic. On the other hand, brick dust molecules have high melting points and a strong crystal lattice. They often have many hydrogen bond donors and acceptors, which leads to strong intermolecular forces that contribute to very stable crystals. Not only are they insoluble in water, but are also insoluble in lipids and many other solvents. Various formulation strategies can be applied to help improve the solubility. The idea is that an enhanced concentration in the gastrointestinal tract will provide a driving force for permeation and absorption through the gastrointestinal walls. The simplest technique for improving solubility is to use different salt forms. Alternatively, looking at co-crystals or screening for polymorphs can help. Breaking up the crystal lattice by preparing an amorphous solid dispersion is another strategy. This can lead to supersaturated solutions and the so-called spring and parachute concept. Cyclodextrins, co-solvents, and lipid-based formulations using oils and surfactants are other approaches. Also, size control strategies such as nanoparticle formulations can also help with dissolution and solubilization. The Biopharmaceutics Classification System, or BCS, ranks drugs according to their solubility and permeability characteristics. Compounds with good solubility and good permeability are in Class 1. Class 2 have good permeability and poor solubility. Class 3 have good solubility but poor permeability. And compounds with both poor solubility and poor permeability are BCS 4. Most compounds coming through pharmaceutical company pipelines are in categories 2 and 4. In the BCS, good solubility is defined as the whole dose being soluble across the gastrointestinal pH range of pH 1 to 8 in 250 ml of water. Good permeability is defined as the drug having greater than 90% human oral absorption. The BCS came out of a regulatory framework. Biowaivers, which avoid costly human clinical trials, can be obtained for drugs in BCS1, provided the dosage forms also have good dissolution. The Developability Classification System, or DCS, is an extension to the BCS and was published by James Butler and Jennifer Dresman. It keeps product development issues in mind and helps scientists to identify rate-limiting steps to absorption and identify appropriate formulation strategies. The DCS moved the solubility line to allow that modern formulation strategies can deliver more poorly soluble drugs. It also divided BCS Class 2 into two categories. DCS Class 2A are for compounds that are dissolution rate limited, and DCS Class 2B are for those molecules that are truly solubility limited. The publication also showed the approximate position for various marketed and GlaxoSmithKline drugs on the DCS plot. The drugs falling in DCS 2A could be formulated using a simple size control strategy, such as micronization, which overcomes the in vivo dissolution rate limitations and allows for complete drug absorption. However, those in DCS Class 2B are truly solubility limited and require an enabling formulation strategy to overcome in vivo absorption issues. This included techniques such as amorphous solid dispersions, lipid-based formulations, and nanoparticulate formulations. This brings us onto the topic of using combined dissolution permeation tools as a strategy for optimizing formulation performance during drug development. Here we see all of the dissolution absorption or dissolution permeation tools that Pion offers. Each of these setups has a donor compartment separated from an absorption chamber and uses the same biomimetic membrane that we used in the PAMPA assay. We refer to these combined dissolution permeation tools as flux setups. The microflux is used for screening in preclinical or early development. The volumes are low and the assay is compound sparing. It can be used for developability or biopharmaceutics risk assessment by evaluating compound behavior and the impact of different dissolution media, such as simulated intestinal fluids, and for selecting ingredients to improve dissolution, solubilization, and absorption performance. The mini-flux adds an absorption chamber into the top of the lid of the mini dissolution tester platform and uses volumes between 130 to 250 milliliter which are representative of gastrointestinal tract fluid volumes. It is used to assess dissolution absorption performance when scaling up from preclinical to clinical development and in the area of children's dosage forms. The bioflux adds an absorption chamber to larger United States pharmacopoeia 500 milliliter vessels and is used to understand absorption performance during clinical development of final tablet and capsule dosage forms using 250 to 500 milliliter volumes. And the macroflux uses an absorption chamber with United States pharmacopoeia 1 liter QC vessels and is used to assess behavior of tablets and capsules in clinical development prior to market launch and regulatory approval. It can also be used to test if generic formulations are equivalent when innovator-approved products come off patent. The method of quantitation for these setups is the rainbow fiber optic platform, and this is described on the next slide. Our main analytical technique for quantitation in the combined dissolution permeation platforms is the Rainbow Dynamic Dissolution Monitor. It is a dedicated diode array spectrophotometer that allows for up to 8 fiber optic UV probes to be inserted into multiple donor and receiver chambers. Rainbow provides us with real-time data acquisition capabilities and allows direct measurements on dissolution, solubilization, and permeation of drugs and drug formulations directly in the vessels without any need for removing samples for offline analysis. Later, we will illustrate its use with several case studies. The gastrointestinal tract lipid consists of the same phospholipids that are used in pamper plates, but here it is painted on the filter of the absorption chamber. The phospholipids infuse through the pores and create an intact membrane barrier, which has been demonstrated to have high electrical resistance, and where the permeability of drugs has been correlated to human permeability. You can see when the technique has worked because it creates a translucent lipid barrier. In the absorption chamber, we use acceptor sink buffer. This is a chemical scavenger that maintains sink conditions and stops back permeation of drugs. Before looking at some flux examples, it's useful to look at the definition of flux. Flux is a measure of transport and describes the amount of API that has passed through a membrane. By definition, it is the net number of moles or the mass of particles crossing unit area per unit time, as seen in the equation. In practice, it is difficult to measure directly inside the membrane. However, it is very easy to measure what appears in the receiver chamber using the rainbow UV fiber optic probes, and the mass change through the membrane can be expressed as the acceptor concentration change over time, multiplied by the volume of media in the acceptor compartment and divided by the membrane area. This is easy to visualize because it means that when you plot the concentration time curve for the amount of drug appearing in the acceptor compartment, it shows that flux is directly proportional to the slope of the curve. The flux can also be used to determine the permeability of drugs according to Fick's first law if the donor concentration is known. Other parameters may also come into play, such as the unstirred water layer thickness and stirring rates. Overall, however, we should be able to determine the effective formulation permeability for different formulation strategies and when we compare different formulations. In the next section, we will illustrate the use of the various dissolution permeation tools with selected case studies. Here is an example of the use of the microflux assay in early formulation development for a poorly soluble BCS class 2 drug, aripiprazole. The aim of this work was to investigate the in vitro dissolution permeation properties of a complex formulation matrix using flux assays, and to evaluate the effect of different formulation techniques on the flux. Various formulation strategies were combined to create amorphous solid dispersions using electrospinning to get thin fiber structures, sulfur-butyl ether beta-cyclodextrin as a solubilizing agent, and citric acid as a pH modifying agent to enhance the dissolution and the bioavailability. Results of a microflux simultaneous dissolution permeation study are shown here. The aripiprazole containing amorphous solid dispersion and a physical mixture of the formulation ingredients were compared to the crystalline API. The concentration time curve of the donor chamber shows that while physically mixing the formulation additives like citric acid and sulfur-butyl ether beta-cyclodextrin significantly enhances the dissolution of the API, the amorphous solid dispersion formulation shows even greater enhancement and creates an ultra-fast drug delivery system. Even though such high supersaturation is not stable, it produces an enormous improvement in the flux across the membrane, as can be seen from the concentration in the acceptor chamber. This is an example of the spring and parachute effect, and approximately 50 times more molecules went through the membrane from the electrospun formulation matrix than from the crystalline form. With the help of the physical mixture, it is also possible to visualize the contribution of formulation additives and amorphization separately. Another thing to note is how information-rich are the donor and acceptor concentration time profiles. With the in situ fiber optic probes, it is possible to capture data very rapidly, as often as every 3 to 5 seconds if we needed to. It is instructive to also look at some literature examples of combined solubility permeability testing. Avital Beg and co-workers conducted a head-to-head comparison of different solubility enabling formulations using a pamper-type platform. Etoposide was formulated in several strengths of 4 solubility enabling formulations: hydroxypropyl beta-cyclodextrin, the co-solvent polyethylene glycol 400, the surfactant sodium lauryl sulfate, and an amorphous solid dispersion formulation. In their pamper measurements, shown on the left-hand side, they have found a decreasing apparent permeability with an increased amount of cyclodextrin, PEG 400, and sodium lauryl sulfate. The right-hand side graphs demonstrates that the solubility increasing effect of these excipients has been compensated by an apparent permeability decrease. They have referred to this as the solubility-permeability interplay. On the other hand, the amorphous solid dispersion samples also provided a significant donor concentration increase due to supersaturation, but the permeability remained the same, despite the increasing load of the solid dispersion and increasing amount of formulation excipients. If we look at the flux as a function of supersaturation level, the amorphous solid dispersion provides an outstanding advantage over the other formulations, as the increase in the dissolved amount provides a proportional increase in the flux and the total mass of drug absorbed. This is because the amorphous drug has a true solubility advantage, and the concentration of molecularly dissolved free drug is much higher than for the other formulation ingredients. In the other formulations, bound or micellar trap drug limits the amount of molecularly dissolved free drug available for absorption. Another example from the literature considers the use of dissolution permeation setup with a CACO2 cell membrane. The group at Setsunan University in Japan demonstrated that dissolution alone does not predict in vivo performance for the poorly soluble drug danazol. They explored a variety of solubility-enhancing excipients and showed that the best overall flux performance was obtained with a formulation with moderate solubility improvement. In summary, when using different formulation techniques, the solubility-permeability interplay has to be considered on overall drug absorption performance, and combined dissolution permeation assays are of great value for assessing in vivo performance. Now we provide another example using one of the PION setups. The group at Takeda performed a pharmaceutics risk assessment on several poorly soluble weekly basic drugs. They used the microflux in a two-stage dissolution permeation assay to see if there was a risk of administering these drugs at elevated gastric pH values. The experiment consisted of a gastric stage on the donor side, followed by the addition of a concentrated fasted state simulated intestinal fluid at pH 6.5. Two sets of experiments were performed, one where the gastric pH started at pH 2, and the second where the gastric pH started at pH 6. This was to represent patients who have been administered proton pump inhibitors or H2 antagonists. The concentration of drug permeating across the membrane and into the receiver chamber was measured over 2 hours and 40 minutes. Selected results are illustrated on the next slide. Three different types of behavior are illustrated on this slide. The graphs show concentrations in the donor vessels on the left and in the receiver vessels on the right. The dark black lines are the experiments for patients with normal gastric pH values of pH 2, and the grey lines are the experiments for patients with elevated gastric pH values at pH 6. The first example is ketoconazole, a poorly soluble wheat base. For normal patients, the whole dose dissolves in the donor gastric pH environment at pH 2. However, when the simulated intestinal fluid is added, there is evidence of moderate drug precipitation at pH 6.5. For patients with elevated gastric pH, very little of the dose dissolves in the donor gastric pH environment at pH 6. Concentration only reaches the solubility in the simulated intestinal fluid once it is added. For ketoconazole, the large differences in concentration in the donor vessel in the simulated intestinal fluid led to large differences in the concentrations appearing in the receiver compartment. The next example is nilotinib, another poorly soluble wheat base. For normal patients, the whole dose dissolves in the donor gastric pH environment at pH 2, but there is rapid and immediate precipitation when the simulated intestinal fluid is added. For patients with elevated gastric pH, very little of the dose dissolves on the donor side. The precipitation and low concentrations of nilotinib in the donor vessels in the simulated intestinal fluid only translated to a moderate difference in the total amount of drug permeating through the membrane and reaching the receiver compartment. Propranolol is an example of a weak base that is soluble across the entire gastrointestinal pH range. The whole dose dissolves in the gastric environment at both pH 2 and pH 6 and remains soluble when intestinal fluid is added. There is only a dilution effect from addition of the intestinal buffer. In the receiver compartments, we see only a small difference between the appearance profiles for propranolol for the two patient groups. The Takeda group were able to use this data, along with several other wheat bases, to show when there is a pharmaceutics risk associated with taking these drugs if the patient has an elevated gastric pH. The results are shown on the next slide. What they were able to show is that if you took a ratio of the areas under the receiver concentration versus time profiles, then this would correlate to the clinically observed Cmax ratios. Hence, anything with a ratio substantially lower than 1 would be at risk of impaired oral absorption for patients with a compromised gastric pH environment. So we see that ketoconazole had the largest difference between flux profiles and also had the largest reduction in Cmax ratio. Nilotinib was moderately affected, and propranolol showed almost no change in flux and did not show a reduction in Cmax ratio or oral absorption. In the last section, before moving on to talk about in silico predictions, I want to show an example of the absorption-driven drug formulation concept using telmisartan as our study drug. Telmisartan was used to prepare amorphous solid dispersions using an electrospinning technology. Both polyvinyl pyridine and the cellulose polymer, HPMC-AS, were selected. Tween 80 was added to optimize the surface tension of the polymer solution, which not only helped the formation of fibers, but also significantly increased the productivity of the process. The scanning electron microscope images of the electrospun samples confirmed the formation of thin fiber structures for most of the formulations. When using HPMC-AS without tween, only electrosprayed particles were obtained. The amorphous nature of the formulations was confirmed by X-ray powder diffraction and DSC measurements. All of the samples were then tested using the microflux equipment. For the small volume flux testing using the microflux apparatus, a media conversion protocol was used, where donor compartments started with simulated gastric fluid at pH 1.6 and then changed to fasted state simulated intestinal fluid at pH 6.5. All 4 formulations dissolved rapidly in the donor media and were completely dissolved in about 30 minutes. They remained soluble in the intestinal stage. The acceptor concentration versus time profile shows that the permeation only started after the media changed to intestinal conditions in the donor compartment. And formulations containing tween 80 show significantly poorer flux values than formulations without the surfactant. This is also illustrated in the bar graph on the right, showing the relative permeabilities. Even though the use of tween 80 produced greater yields from the electrospinning, the flux assay results prompted us to select the 2 formulations without surfactant for the next step, final dosage form preparation. The polyvinyl pyrrolidone and HPMC-AS solid dispersions were moved to the next stage of final dosage form preparation. Tablets were also prepared with either sorbitol or mannitol as fillers and sodium hydroxide as pH modifier, as these excipients were present in either the marketed innovator product or in some generic formulations. So 4 different tablet versions were made, as shown by the table, varying the type of polymer and type of filler in the tablets. These tablet samples were then studied using the macroflux large volume dissolution permeation apparatus, and we wanted to see if our formulation approach could match the performance of the branded product from Boehringer Ingelheim, known as Mycardis. The macroflux system, coupled with in situ fiber optic concentration monitoring, has been used in this final investigation of the tablets. A 2-stage gastric to intestinal media conversion protocol was used in the main dissolution vessel. The donor data on the left side shows a delay in disintegration and dissolution for the HPMC-AS mannitol tablet, but it also reached 100% dissolved after about 60 to 70 minutes. The permeation to the acceptor started after the media conversion, and the flux was constant in the studied time frame. The amount of permeated drug in the acceptor compartments gives a quantitative comparison of the formulations. Both sorbitol-containing formulations performed better than the formulations containing mannitol, regardless of the polymer type. The HPMC-AS-based tablets with mannitol provided the lowest amount of permeated drug in the whole study. The results of these tablets were also compared to the branded innovator tablet. The branded version of telmisartan is known as Mycardis and is produced by Boehringer Ingelheim. The bar graph on the right, displaying the relative permeabilities, shows that the 2 sorbitol-containing tablets performed similarly to the branded Mycardis formulation. Hence, both sorbitol-containing tablets containing either PVP or HPMC-AS can be expected to perform equally well in human in vivo studies. In the last section, we will cover the use of our in silico tool that works with dissolution permeation data to evaluate oral drug absorption. The software is known as Predictor, and it can be used to determine maximum absorbable dose, oral fraction absorbed, BCS class, and rate limiting steps to oral absorption. The GUT framework stands for the Gastrointestinal Unified Theoretical Framework and is recreated here from Keo Sagano's book, Biopharmaceutical Modeling and Simulations, Theory, Practice, Methods and Applications, published by Wiley in 2012. The GUT framework comprises a system of equations which model the processes of dissolution, precipitation, and absorption. In terms of parameters needed for making a prediction, the gut framework requires the input of certain measured physicochemical properties and constants in order to appropriately calculate dissolution and precipitation rates and the permeation of a drug, and to ultimately determine the oral absorption. I don't intend to go through all the equations, but use it to illustrate the complexity of all the interconnecting processes. Interestingly, in vitro flux can be used as a surrogate for many of the dissolution permeation processes shown here. So using flux results derived from Pion's flux assays allows the substitution of many of the equations feeding the dissolution and permeation parts of the gut framework. Hence, it is possible to determine the amount of a substance permeating the intestinal wall using only the value of flux supported by the solubility and dissolution behavior observed in the donor vessel, providing the source assay is designed to emulate intestinal conditions as closely as possible. This then gives us the ability to determine the maximum absorbable dose and the oral fraction absorbed for the formulation. Translating in vitro results to in vivo relevant results is performed using Pion's predictor software program, which is a new data handling and prediction package that works with dissolution permeation data from Pion's Rainbow instrument. The total mass absorbed in vivo can be written as the integration of the in vivo flux multiplied by the surface area of the human intestinal membranes, multiplied by the transit time in the human intestine over which drug absorption can take place. This is the maximum absorbable dose. The fraction absorbed is then the total mass absorbed divided by the administered dose and multiplied by 100 to get it as a percentage. However, what we need to be able to do is to take our in vitro determined flux values in our dissolution permeation assays and apply certain scaling factors to be human in vivo relevant. These include things such as corrections for the in vivo absorption barrier properties, including unstirred water layer thickness, scaling to the available intestinal surface area based on the drug's permeability, considering dose clearance from the donor side of the in vitro system due to the smaller membrane surface area, and making adjustments for intestinal transit time. In the next slide, we'll see an example of in vitro flux scaled to in vivo oral absorption. Here we see the acceptor flux profile for a formulation of aripiprazole. It is imported into the predictor software, and the species of interest is selected. In this case, the default species is a human. The scaling factors mentioned previously are applied, and the output is the graph on the right, showing the oral fraction absorbed versus intestinal transit time. The predictor software model has been validated against marketed formulations with published human oral absorption values. The data shows a good correlation using different flux apparatus setups, and most predictions are within plus or minus 15% of the published oral fraction absorbed. Several compounds were also studied at different dose levels. For example, for carbamazepine and celecoxib, the fraction absorbed was lower for the highest dose levels, thereby illustrating that solubility is likely impacting on the overall extent of oral absorption for these compounds. Another outcome of the dissolution permeation experiments and the fraction absorbed calculation is that we can also determine the dose number, dissolution number, and permeability number for the drug in the formulation. These are dimensionless numbers that tell us about the dissolution, solubility, and permeability behavior. The dissolution number is the residence time of the drug in the intestine divided by the time for dissolution. If the number is greater than 1, then the whole dose can dissolve. The dose number indicates if the drug is soluble in the intestinal fluid volume. A dose number of 1 or less means it is completely soluble. A dose number of 2 will mean 2 intestinal fluid volumes are required to dissolve the dose. A dose number of 10 would mean 10 intestinal fluid volumes are needed. The permeability number is the permeability rate constant multiplied by the intestinal residence time. High permeability numbers translate to more complete absorption. We can display any two of the dissolution number, dose number, or permeation number in a three-dimensional graph against the oral fraction absorbed and identify rate-limiting steps to oral absorption. In this example, we show results for itraconazole formulated as Sporanox capsules and as Sporanox oral solution. The capsule contains beads of itraconazole as a solid dispersion. The oral solution contains itraconazole in solution with a large proportion of cyclodextrin. The solid dispersion is only about 40% absorbed. The graph shows that if we can increase the permeability number or decrease the dose number by improving the solubility, then we will walk up the surface and improve the overall oral fraction absorbed. The oral solution illustrates how this can happen. The permeability number is higher and the solubility has improved with a lower dose number. The oral fraction absorbed is approaching 70% for the oral solution. Finally, we can also highlight the rate-limiting steps to oral absorption. For example, is it dissolution rate limited, permeability limited, or solubility permeability limited? In this case, both itraconazole formulations exhibit solubility permeability limited oral absorption. Knowledge of these processes can help formulators identify what aspects they need to address to improve their drug formulations. In conclusion, these are some of the things we've learned about combined dissolution permeation strategies. We have looked at the impact of biorelevant media on drug performance and seen the importance of evaluating excipient-drug interactions via the solubility-permeability interplay. We have looked at examples of two-stage gastric to intestinal dissolution permeation testing on biopharmaceutics risk and for developability profiling. And finally, we have combined experimental dissolution permeation data with mechanistic modeling to provide a more quantitative aspect to oral drug absorption and to also help formulators identify the rate-limiting steps to oral absorption. Thank you for listening. I will now hand you back to the moderator for questions.
Thank you. Thank you so much, Carl, for your engaging presentation today. We truly appreciate the time and expertise you shared with us. It was a pleasure having you as part of the webinar. We will now move on to the last part of today's webinar, the question and answer session. As a reminder, to ask a question, you just need to simply head to the engagement window to the left of the auditorium and click to open the Q&A window. So to kick off this final portion of today's event, the first question I have reads, you showed coating of phospholipid onto the membrane filter in one of the slides. How robust is the membrane to different dissolution media and to various formulation ingredients?
Yes, thank you, Olivia. So yeah, I can answer that question. So the membrane, I think we saw in the slide, we coat the filter. It's a PVDF filter. We coat it with a mixture of phospholipids. The phospholipids infuse into the pores and they create an intact membrane barrier. And that membrane barrier has been confirmed by TEER electrical kind of resistance measurements that is the main barrier to movement of molecules, and it's actually very robust to different conditions. So different types of media, for example, we've used fed state simulated intestinal fluid as well as the fasted state, and it seems to be fairly robust to different formulation ingredients as well. There may be some concerns around high oil and surfactant content with lipid-based formulations. But the other thing we can do is we can usually tell from the data, the fact that we have the fiber optic probes, we can capture a lot of data very frequently. If there is any issues with membrane integrity, we would often see a break in the data, and that would tell us that something's happened. Usually we'll have enough, there'll be good data that we can use up to the point of where that might happen if it does happen, and it doesn't happen very often. And the other thing we can do is we can always put negative markers into the donor sites. So, for example, we could use a dye molecule or something that has a strong UV absorbance but should have no permeability, and therefore we would see if that was able to get through the membrane. And we can do that in, either we can dose it along with the compound and the formulation, or we can set up a kind of placebo flux pair where we would just have the dye in with the formulation ingredients to see what would happen. So I would say on the whole, yes, we've demonstrated that it's very robust to a lot of conditions.
That's great to know. Thank you, Carl. I've got another question here that says, can you say something about dose levels? Do you use whole tablets or capsules in the microflux?
Yeah, I mean, I can take that question as well.
Yeah, thank you.
So in the microflux setup, so this is the small volume apparatus. We don't tend to use whole tablets or whole capsules in the low volume setup. We want to try to be dose appropriate to intestinal fluid volumes. So, for example, if we assume that the intestinal fluid volume is around maybe 130 milliliters in the gastrointestinal tract, we have a 20 milliliter volume, so we would scale it 20 divided by 130. So a 100 milligram tablet in practice would probably end up using around 15 milligrams of API to be kind of dose proportionate to the intestinal volume. For the larger scale apparatus, we can indeed use whole tablets and whole capsules.
That's great. Thank you, Carl. The next question I have says, you showed that the flux data can be used to predict oral fraction absorbed and other parameters in your predictor software. Can flux data be used with the physiological based pharmacokinetic modeling software, PBPK, supplied by the major companies in this field, for example, Simulations Plus or the SimSip Satara?
Do you want to take this question, Berlin, or should I take it?
Yeah, it's fine. I can try to answer that. So our modeling platform is really a unique modeling platform that basically builds on our flux data as the primary input, and we use several other inputs, of course, solubility values. But in general, that's what we use as an input, and that's what we use to predict the absorption. So we are very much focused on the absorption side of the story, while general PBPK modeling platforms, they are like a broader platforms covering distribution, elimination as well. But to answer the question a little more directly, we can generate in vivo effective permeability predictions that can be used as input parameters for many of these software platforms. So that's the I guess the best parameter to use to further process the data in the PBPK platforms.
That's great. Thank you, Valent. Another question here says, when you describe the different flux setups from small scale to large scale, you mentioned that the largest apparatus has been used to determine bioequivalence. Can you describe this, and is the data accepted by the regulatory authorities to support biowaivers?
Yes, I can take this. So the macroflux device, yes, we have been using it to compare formulations from developed by generics companies versus innovator products, and essentially we just want to compare the flux profiles to see how similar those results are to the marketed innovator product. And we've been showing that if the ratio of the flux data is within a band of like 80 to 125%, then we would presume that those products would demonstrate bioequivalence. And so, yes, so we've done that. And I think the macroflux setup is, it's the closest setup to the QC dissolution vessel, but we don't believe that just dissolution data alone is always giving the best comparison of product performance. So this is why we think it's better actually, or more useful, to also look at the flux data. In terms of regulatory authorities, so I mean, the Chinese FDA have a lot of our instruments, and the Chinese FDA have been publishing a lot on comparison of flux profiles, as I say, of generic products in China compared to innovator products, and getting quite a bit of success. So we would suggest that people can probably use the flux information as supporting type information in dossiers or packages. But we're yet to see the FDA or any other regulatory authority kind of officially approve this format. But I think progress has been made, and especially with the Chinese FDA.
That's great. Thank you, Carl. I've got another question here that says, would there be a scenario that the dissolution difference was observed in the donor side but not reflect in the acceptor side flux profile?
Do you want to take this one, Blint?
Yes, that is an absolutely possible scenario. Just because certain excipients or formulation strategies are increasing the dissolution or increasing the overall solubility of the compound in the donor media, that does not mean that the higher concentration or all the molecules will be available for absorption. It really depends on what is the rate limiting step of the absorption. For example, if it's a transmembrane epithelial membrane permeation limited case, then micelles or cyclodextrin will not necessarily help the absorption, even if it provides a higher concentration. While if it's an antivoto layer rate limited permeation, then those formation strategies can actually be powerful. So it really depends on the whole system, on what is the or how the compound is being absorbed.
Thanks, Belen. Thank you. I've got the next question here that says, what can you tell us about the dose concentrations employed? Does the microflux methodology require intact tablets/capsules or modified formulations?
So I think that's the second that question's come up before. So I think we answered it when we said we do kind of dose. We scale the doses appropriately to the intestinal fluid volumes. So I think we already answered that question.
Oh, okay. Yeah, perfect. Thank you. Next question here says, which pharmacopoeia general chapter can we refer to in studies for study?
I mean, there is no pharmacopoeia general chapter, I think, around flux. As I mentioned, the Chinese FDA are publishing on this. They have our technology. They're publishing quite a lot, but I don't think there's any general pharmacopoeia chapter that you can read on flux approaches.
That's okay. Thanks. I'll move on. Next question here says, is it possible to quantify two or more components with similar absorption maxima?
Do you want to take this one, Blint?
Yes. So Carl had mentioned that our UV technology can actually deal with two or even three absorbing components in the same solution. If they have a similar absorption maxima, then that really depends on how the overall shape of the UV spectrum looks like. Even with the same absorption maxima, if the shape is different, then the second derivative spectrum will be different. And if they have different x-axis crossing points, what we call zim points, or if they have an overall different shape, then one of our two methodology of how we deconvolute the overall absorption spectrum is likely to work. Of course, both mathematical approach has its own limitations, but in many cases, if it's two components, then one or the other tends to work fairly well. We have seen situations when compounds are like very similar, even from a structural perspective, like methylene and propylparaben, for example. And of course, they have a very similar spectral shape, and that's a bit more challenging for the method. But typically, if it's like more kind of structurally different compounds that has some significant differences in the UV spectrum anywhere, not just at the absorption maximum, then we can typically deconvolute those.
Great, thank you, Villain. I've got another question here that says, typically the acceptor side of the flux setup is filled with a high concentration surfactant solution to create sink condition. How do you prevent osmosis from taking place?
I mean, I think this goes back to the kind of membrane integrity discussion, and I think we've demonstrated. So I think I gave quite a long answer before about membrane integrity and the ways that you could evaluate that. And sometimes you may see a break in the data from the UV fiber optic data, or if you wanted to test a particular setup with a particular media and particular formulation, you could use a negative marker dye or something like that. So I think it's the same thing. It's membrane integrity has been tested, and it's robust under a lot of different conditions.
That's great to know. Thank you, Carl. Last question here that I've got. It says, please can you suggest the best predictor software from in vitro dissolution profile to in vivo performance of the formulation?
Do you want to take this, Belen?
Yes. So I guess it's it's really a hard question from the perspective that we cannot really suggest. I mean, there are numerous systems that claims to perform well in many scenarios. Our belief is that just by the in vitro dissolution profile, especially for BCS class 2 drugs, it is very difficult to make accurate in vivo performance predictions because of all the factors that Kat have mentioned, because it really depends on the excipient composition, on the formation strategy, on the particle size, for example, for a non-ionized formulation. So, and just by the dissolution, you cannot really see those effects, and therefore the dissolution permeation setup is the tool that can give that extra piece of information that many of these prediction software that only uses dissolution as input will miss. So I guess I cannot really suggest any of those platforms. I mean, though I'm sure they work well in many scenarios.
Great, thank you, Belen. Well, I'm afraid that is all we have time for today. I'd like to give a huge thank you to our amazing guest speakers, Carl Box and Belen Cinco, for sharing their knowledge and expertise. And of course, thank you to all of you who joined us today. We hope you found the discussion insightful and that you walk away with new ideas and perspectives. If we weren't able to get to your question today, don't worry. We'll be following up with the answers after the webinar. And if you have any additional questions after the session, please feel free to reach out to me at the email address editor@selectscience.net. Certificates of attendance will be automatically emailed to all live attendees a few days after the event. For those watching on demand, a certificate can be requested by emailing us at the same email address. The on-demand recording will be available in just a few days, allowing you to revisit the webinar or share it with a colleague or friend. Once again, thank you to everyone for attending. Stay tuned for updates on future webinars, and we hope to see you again soon. Take care and have a wonderful day ahead.
Thank you.
Thank you.
Thank you.